mostly fluid compartments and also give structure, enable sliding, and give resistance to mechanical shear. And furthermore, they also define the outer appearance of the whole thing. So there was a definition of fascia which has been introduced at the first fascia research conference at Harvard 2007, and this includes a definition that is a three-dimensional network throughout the whole body and includes all morphologies which are shaped by tensional loading. So this includes the superficial fascia, deep fascia, it includes aponeurosis, organ membranes, and joint capsules, even tendons and aponeurosis. So if you look at this 700-year-old lady from Peru, I admit she looks a bit rusty, but still you can see that she has a posture and you can imagine her face. To me this is quite impressive because basically what you see is the shape-forming effect of fascial tissue. So let's go into these physiological properties of fascia. This is the classical assumption of muscle contraction and relaxation, and this gives elongation and shortening of the calf, and the tendons more or less passively give the force generated by the muscles to the bones. However, this classical hypothesis is wrong. So new results of Kawakami show that the muscle fibers contract in the calf almost isometrically. So everything done in shortening and elongation is done by fascial tissue, connective tissue, which not only transmits the force to the bones, but also modulates and acts as a recall spring, which buffers energy and modulates energy. So if you look at Oscar Pistorius, who wears resilient lower leg prosthesis, and he actually qualified for the Olympic Games. So there is evidence that the elastic recoil is very important for our locomotion. So even if he lacks a significant part of his lower leg muscles, he can run as fast as other athletes with their complete muscles on the lower leg. Several species use this elastic recoil function of the lower legs as their primary mode of locomotion. So this has been shown not only in wallabies but also in other species. So the elastic energy storage capacity is not limited to the lower leg. And so these are data of Andrew Franklin Miller, who couldn't attend the meeting unfortunately due to personal reasons. And he showed that if you do a straight leg raise, then the force is not only, tension is not only transmitted to the posterior part of the leg, but also, and even twofold, to the iliotibial tract, and surprisingly to the lumbar fascia as well, which is a very huge aponeurosis covering the whole body basically. So there was a Nature paper back in the 80s, and they showed that women of some African tribes carry heavy loads on their head without an additional energy consumption. So this means that the energy is not supplied by more muscle work, but has to be stored by some other mechanisms. The authors didn't know, of course, what it is, but they speculated that it has something to do with elastic recall. And our group, Achuton, he did mathematical calculations with Lagrange calculations, and he found out that this elastic recoil of the back of this thoracolumbar fascia is a big storage for elastic energy and can help in this energy saving mode of walking. So let me say some words about fascial contractures on a physiological and a pathophysiological point of view. This is a slide of a lumbar fascia of a 19-year-old guy, and you see the crimp formation of this collagenous tissue, and you also see some stained dots. These stained areas are stained for myofibroblasts by staining for alpha-smooth muscle actin, and myofibroblasts are contractile cells which are found inside fascial tissue. So we also did a quantification of these stained areas, and we found out that there is a huge variance, not only in between individuals, but also in between different locations of the body. So, what are myofibroblasts? Myofibroblasts can proliferate and differentiate from fibroblasts by triggered by mechanical tension or by cytokines like tissue growth factor beta 1, which is involved in inflammation. So, if you look at a physiological point of view, then we did some studies in an organ bath with controlled conditions. We had controlled temperature, controlled oxygen, pH, and so on, osmotic conditions. And then we get this time versus force registration. And this is a sample of a fascial tissue bundle which exhibits a significant contracture in the time scale of several minutes, similar to smooth muscle cell. So, mepyramine is the trigger substance which we used, and mepyramine is an agonist on histaminic receptors. So what is the mechanism behind? So, this is the biochemical pathway of myofibroblasts contraction. You can see that there is a rho kinase dependent pathway which inhibits myosin dephosphorylation and leads to sustained contractions. This pathway, interestingly, is calcium independent, unlike all other tissues, unlike skeletal muscle, unlike thrombocyte contraction, unlike smooth muscle. So what is the standard pathway is calcium calmodulin activation of myosin light chain. And so there is a study group around Boris Hinz from Toronto, and they found that myofibroblasts have cyclic oscillation spontaneously, so similar to cardiac myocytes. And this spontaneous oscillation can be modulated by, let's say, in vitro by tapsigargin, which is a calcium ionophore, and so the beat displacement, which represents the force after this calcium oscillation, can be abolished. So tapsigargin is not available for clinical use, it's toxic. So if you find an alternative, you can get very rich, because the cosmetic industry is interested in stopping wrinkle development and stuff like this, and this would be one key mechanism, obviously. So if you look at a myofibroblast at work, it doesn't only run through the tissue, but it also remodels. And the authors estimate that roughly 1 centimeter contraction per month can be done by this in vitro forces. So, myofibroblasts are involved in several pathologic contractures like Viking's disease, morbus Letterhaus, frozen shoulders, and frozen lumbars as well. So, let me comment on fascia and pain syndromes, and there are quite good data about lumbar fascia, that's why I go into this field. So this is a well-accepted hypothesis for the development of low back pain. If you look at the main components, this is the spinal column and spinal muscles, where coordination and activation is disturbed during this mechanism of back pain. So we looked at an MRI scan, and we found out that the lever to the facet joint